Hello, in this class I'll be teaching you on DNA analysis. So we'll be working on DNA analysis on the general note. In the previous class we talked about getting familiar with Genious environment. So I think with that you should be able to get familiar with whatever we'll be doing here, and then you'll be able to flow with me. Now we'll be working on DNA analysis on the general note. Now follow me very carefully as I take you through the process of analyzing DNA data. The first thing you do is create a folder. So you go to the file option. As you can see, you create a folder and you rename the folder. So we're working on DNA analysis, but mind you, we are working on a project which is SQL cell. From the beginning we have been working on SQL cell analysis on the database and internal too. So that's the data we'll be using. So and all the assignments you'll be given, it is expected of you follow the same process to be able to work on your assignments perfectly according to the way you are being taught. So now you rename this folder. So we're working on DNA analysis, but then on those SQL cell data. So what you will do is rename the folder to so as you can see, here is it, SQL cell research project. SQL cell research project. So what we'll be working on right now on those data, like SQL cell data, they're all gonna be under this folder. As you can see, SQL cell project. So the next thing you will do is import the files, the data you downloaded from NCBI database. So you click the file option, go down to import. So then you click. So you look for the folder you saved. Those data you downloaded from NCBI. So I named my own to be SQL cell. SQL cell. So here is it, SQL cell. So you click it and you find both data. So you can highlight everything here, so and then import everything to the Genious software. So the Genius software will ask you if you keep sequence separate or create sequence. So keep sequence separate. So you have it here. Now this is the Homo sapiens mutant metaglobin. This is the mutant file of the SQL cell, while this is the normal file of the SQL cell. So the next thing we do now is we work on the DNA analysis. So once you're working on the data, you're working on research, first thing you do after importing your file is that you do the DNA analysis, which we are about to embark on now. So just follow me very carefully as I show you how to work it out. So presently now I will be working on the normal gene, while I will give you as an assignment to work on the mutant gene. So just follow me very carefully. We're about to embark on the DNA analysis using these sequences. So you will click on the human beta globin region on chromosome 11. So I will click on it, and then I want to understand the nature and the look, the DNA makeup of this sequence. I want to understand it, and that is DNA analysis for you. So now this to give you a general note and understanding of what you want to work on. You understand? So you downloading the data from the database, you just need to understand what that data is all about. You need to know if it is DNA, you need to know if it is RNA, you need to know if it is protein, you need to confirm. And there are a lot of things that are attached to DNA analysis that you need to understand before you can begin to go deep into working on that data. Maybe go deep into working on gene expression, go deep into understanding the functions of the genes in that sequence. or you want to understand the annotations, you understand, proceedings on the sequence, you want to understand the annotations, you want to understand the informations that are found in the sequence. So all you need to do is, the first thing you need to do is embark on DNA analysis. So if you can be able to do that, you have a better study background on what you want to work on. So just follow me very carefully as we keep moving. So I just click on the human beta globin region on chromosome 11, and immediately I click it brought this. So this region is the region that show whatever you click here, it show you everything. It shows you everything, including all the nucleotide bases from number 1 to the last number. Now as you can see, the total number of the sequence here is 73,308 nucleotide bases in this sequence. So that's the first thing you do. The first thing you do is what? Understand the total number of nucleotide sequences in this in this sequence. You need to understand the total number of the nucleotide bases in this sequence. That's the first thing you do. So immediately click it to give you here. So it's starting from number 1 to 73,308. So this is the last part of the nucleotide bases in this sequence. So the next thing you do is you zoom, keep zooming. Yeah, so these are the sequences starting from number 1. So you can then is in a wrap form. So you can unwrap with this place, you can unwrap. So you have something like this, and you can this it, you can zoom it so that you understand what you have, and you can wrap it again so that you're able to have full sequence view. So as you can see, so this is 1, this is 10, this is 30, this is 50. So that means from here to this place is 50. So from here to this place, the whole nucleotide sequence, basis there is 80. So if you keep going, so from the number 1 to this place, we have about 54,870 nucleotide bases. So you keep going. So this is the last nucleotide base in the sequence, which is what? And the last one is 73,308 nucleotide bases in total in the sequence. So that's that. Now mind you, what we are viewing is under sequence view. So you are actually viewing the sequence. You're actually viewing this particular sequence. So that's the first thing you do. Now mind you, in case you uploaded and you didn't find anything, so you can just click it and then click the home icon here to be able to see other options. Now the next thing you do is understand what kind of nucleotide sequence you're working on. Understand what kind of nucleotide sequence you're working on. You're working on the DNA sequence, you're working on the RNA sequence, or you're working on the protein sequence. So mind you, the difference between DNA sequence and RNA sequence is the difference in the base they share within themselves. Like RNA have uracil, which is the change of timing to uracil. So in DNA sequence, for you to be able to recognize DNA sequence is when you see T in the sequence. So that means it's a DNA sequence data. Now once you find U in a sequence, it's telling you that it is a RNA sequence, RNA data. So the next thing you move on to is understand the graph. You understand the graph. So this is the option for graph. So if you click it, you can remove it, and you have your full intact sequence without any gap. So as you can see, you can zoom, you can reduce, just like that. So you can add the options graph on it. So as you can see, so we have all the graph here. So in case you unwrap, you still have the graph this way. So you can just move it, move it. So you have all the sequence. Now there's a significance to the graph. Let's wrap it again. There's a significance to this graph. So this graph is telling you a particular message, is giving a particular message. Now we have a total number of 4 kinds of nucleotide bases in every nucleotide sequences. So we have ACGT, but I want you to understand something, that for you to understand this graph, you need to understand the significance of each database in the sequence. So first of all, you need to understand the stability of each nucleotide bases in the sequence, because that's how you will understand the graph. Now listen very carefully. A is not stable in a sequence. Now C is stable, G is stable, while T is not stable. Now this is the essence. A and T shared a double bond. You understand? So, but G and C shared a triple bond. Now this, I'm talking about hydrogen bond now. So G and C shared, have triple bond, so it is well stable. You can search that on Google, like the stability of nucleotide bases, so that you'll be able to understand what I'm trying to tell you. So AT share double bond, so it's not stable, while GC share triple bond, so it is stable. Now the significance of the triple bond found in CG is trying to tell you that the entire nucleotide sequence can only be stable if the GC content, the GC stability content in the sequence is high. So if the GC stability content in the sequence is not high, then the sequence is not kind of stable. It's not stable. Now that means there's probability that the sequence can easily be dominated, be affected by foreign invasions. Are you getting it now? So, but if the AT content in the nuclear sequence is low, while the GC content is high, so it's telling you that the nuclear sequence is stable. So the essence to that is that the sequence have high capacity to withstand, to resist any foreign invasions. So now this graph is giving a direct meaning to the content of these nucleotide bases. Now follow me very carefully. As you see from here to this place, it's telling you that this place, the graph is kind of high, and for it to be high, it's showing you that G is found. As you can see, G is found. Now this is telling you that this region is highly stable. That's the essence of the graph. Is what? Highly stable. Now you see from here to here, as you can see, it keep reducing, keep reducing. As you can see here, kind of high. So this graph is trying to make you understand the stability of each nucleotide sequence. The stability of each nucleotide sequence, as you can see. So G is there. So as you can see, the graph is high here. C is high in this place. So this place is extremely low, right, because of the T. So a little bit high because of the C, low, high, high, low, low, high, high. As you can see, keeps going just like that, like that. So that is the essence of the graph. Now, just for you to understand that this sequence is a sequence strand of the structure, the double helix structure of DNA. This is just a sequence strand. Now, so you can actually impute the double strand of the DNA. So all you need to do is click complement. You click the complement. So this is a single strand, but this is the double strand of this nucleotide sequence. So but majorly what you downloaded is the single strand. So but you can actually deduce this double strand using the Genious application. Now this for you to understand that in double strand DNA we're talking about the complements of each nucleotide basis on the first strand. You understand now? So as you can see, we have G here, then we have C, which is the double strand of this nucleotide sequence. Now we have A here, and then we have T, we have A, we have T. So this should tell you that G pairs with C, while A pairs with what? With T. So that means that means G, which is guanine, pairs with C, which is cytosine, and while A, which is adenine, pairs with T, which is thymine, and so on and so on and so on, like that. Just like you can see, CG, TA, TAAT, CG, GC, and things and continuous like that to the end. So now this actually how the double helix structure look like in the body of living organism. Alright, so we'll be working on translation right now. So remember I told you that in translation we're talking about RNA being translated to protein, and if we talk about protein we're talking about amino acids. So and in your assignment I believe you understand what amino acid is all about. So and remember I told you that 3 nucleotide bases made up a codon, and this codon codes for an amino acid, which is actually a protein. So that's the gene expression in a way. So but we're going to be talking about gene expression, but then we'll be looking at how these amino acids actually code for specific functions and structure. That is later on in this bioinformatics course. So but right now let's look into how this Genious application can actually deduce each codons and a protein in this DNA analysis that we're working on. So for you to work on translation, all you need to do is click here, as you can see. So we've talked about complement, so we're going to translation. So just click translation. So as you can see, presently on your screen, this is the 3 nucleotide bases that actually code for an amino acid E, and this E signifies glutamic acid. So this is actually an amino acid. Now we have TTC, and this TTC codes for symbol, and the symbol is F, which is an amino acid, and the amino acid is phenylalanine. So and then so on. And TCT code for the symbol S, and S is serine. S is serine. So we have CCC code for an amino acid with symbol P, and P is what? Proline. You just keep studying the nucleotide sequence, and you keep on finding the amino acid, the codons, those 3 nucleotide sequence, and then you keep on finding the stop codons. To talk about graph the other time, you can choose to remove it and then put it back. Then
GC content. Now this is a graph for GC content, and that's significant to this graph. Now as you can see, the lines here, we have green lines, and we have blue line. And I told you the other time concerning the GC content and AT content. I want you to understand that if we're talking about GC content, we're talking about blue lines. So, and then if we're talking about AT contents, we're talking about green lines. So now it is for you to understand that this graph is making us to understand the express level of the GC content and the AT content. GC content is not expressed here, either AT content is expressed. As you can see here, AT is green, content for selected sequence. Now, so that means this is AT content, which is the green line. So that means it's more expressed. That means it's above, blue line, which is GC content. So that means entirely this region from here to here. So as you can see down here, you see, you keep seeing green line. So that means you look at AT, just like that. Keep seeing it. See, so you see, the green line is more expressed. So that means this is trying to make us understand that in this region, C and G is more expressed, just like you can see. You can see G here, G, C, another G, C, another C, just like that. So that's the essence of this. So you can just remove it. Now there's something that's very significant here. Just watch. So that is the stylized DNA helix. Now this stylized DNA helix is just trying to make us to understand or see to the animated form of double DNA element, double stranded, so just trying to make us to understand the DNA helix, which is the double stranded DNA structure. Yeah, so then just click it and then you find something like this. How wonderful. So there's, go back to the first, place. This is 1, 4. So this is where the sequence actually begins. Now as you can see, this is actually a DLX helix, you understand, a double-stranded DNA structure. So now each of these nucleotide bases have a color, significant color that are specific to them. So we have G. Now this G is what orange. What color is it? Is orange color. So that means in the DNA structure, this G is orange color. So just as you can see here, is orange color. Now this blue you are seeing is actually the complement of this G on the helix structure. You understand? Double-stranded, DNA structure. You understand now? So all you need to do for you to really understand is impute the complement. So just come here and impute the complement and go back. So just like you can see now. So G is orange, while C is what? Blue. So here's it. Now look at this region. A is what? Red. Look at it. A, which is the double strand of this sequence. You understand now? Now the first strand is what? Is T, which is what? Which is blue. So you can actually view the animated form of this DNA helix. I mean the moving form. So you can just click this animated and then you see it moving. Another thing we do is, let's look at the next thing, which is by clicking this icon here, as you can see. So this gives us a brief summary of this particular sequence we're working on. So what you need to do is begin to understand what this data is all about. So what this statistics is all about. So just listen very carefully. Now all this signifies each nucleotide basis. Now A is adenine, C is cytosine, G is guanine, T is thymine. So that means there are about 22,068 of adenine in the whole of this nucleotide sequence, and it's about 30.1% of the whole sequence. So the same thing with cytosine, guanine, and thymine. So the GC content is trying to make us to understand that the extent or the amount of GC, which is guanine and cytosine, in the whole of this nucleotide sequence. So that means you deduce, or you get the total number of this GC content by adding the number of this nucleotide base, which is cytosine, and that of guanine. So you add it together to give you the total number of GC content and the percentage. So the percentage is actually gotten from adding this the percentage of guanine and cytosine together. So that's one thing about that. Now we have rough thing, which is about 81.1 degrees Celsius. Now this rough thing means temperature, and this temperature is the amount of the temperature that is needed to denaturate this nucleotide sequences. So denaturate means the process by which you denaturate a nucleotide sequence or a DNA to get a single strand sequence or DNA. Sure, you understand what I'm trying to say now? So now we have amino acid and codons. So now this is actually telling us the statistic of each amino acid in this nucleotide sequence. So this is telling us that A, which is alanine, is getting 966 terms, you understand, in the whole of this nuclear sequence, according to the codons that have been predicted. So that means this alanine is like 4% of the whole nucleotide sequence. So, and they do the same for all amino acids here. So all these are amino acid. These are the numbers at which they occur. These are the percentage. Now we have that of codons too. After those codon, so we have codon, the percentage and the frequency. So the GCA codon, which actually code for A, which is alanine. So this is actually the amino acid. It is about 37% of the whole of the nucleotide sequence. So it exists. The existence of it is about 37%, and it exists about 357 times in the nucleotide sequence. So, and the same thing for all nucleotide for all codons and the amino acid which they code for, and the percentage and the number of time or frequency they exist in the nucleotide sequence. So I think that's the last part of it in this aspect. Now the other thing I will try to show is the dot plot. So the essence of this dot plot is for us to understand similarities of sequences that exist between two sequences. So, and then they are being deduced by dots. So just like this, dots here, this dots here, this dots here. So this dot here is actually making us to understand that there's a particular nucleotide sequences or nucleotide bases that exist in this area, that is being compared or seen between the two stranded. So this is actually telling you that there is a nucleotide sequence here that have a similar resemblance. So they are similar when it comes to the relationship between the two sequences. So, and then we have text view. So this is the text view of this whole sequence, and then we have the info. So this is trying to make us to understand that the molecular type is DNA and description is hemoglobin beta region chromosome 11. So this is all about DNA analysis.